Hello, everyone, and welcome to URDC 2021. Thank you all for joining in and watching our research presentation, and I hope you all enjoy it. Our current research is on the effects of GPR1 activation on progesterone receptor gene expression in meningioma cells. My name is William Yost, and this is my partner, Zachary Matheny, and a big thanks to our research professor, Dr. Hankins. Meningioma and glioblastomas are two of the most common central nervous system tumors. Meningiomas, which arise from the meninges that cover the brain and spinal cord, occur twice as frequently in women as in men. And glioblastomas are almost always fatal tumors that arise from astrocytes that form supportive tissue of the brain as well as forms the blood-brain barrier. Glioblastoma incidence in men are 1.5 times as likely as women, and this is in contrast to the meningiomas. Some background and important information to be aware of is because of these facts, it has been postulated that steroid sex hormone plays a key role in brain tumor biology. However, the full mechanisms are not well understood yet. It is important to note that to date, most investigations have focused on classical steroid hormone receptors, not receptors that are bound to the cell membranes. We are particularly interested in the membrane progesterone receptors.
It's just some more important information is that our lab has previously demonstrated that membrane-bound G protein coupled receptor GPR1 is expressed in the cell line that we worked with, which is CH157 found here, human meningioma cells. This was a gel that was previously done, and the main focus here are these classical nuclear estrogen receptors, alpha 36 and beta, as long as with the GPR1 that we're focusing on. And this GAPDH is more of just a housekeeping gene, and it's just to show equal loading. Here are some pathways activated by estrogen through GPR1, where estrogen readily enters through the plasma membrane because it is a cholesterol derivative, where it can split off into two typical pathways that we know of, at least here, which is more of a classical nuclear pathway, where it enters directly into the nucleus, where it can act as a transcription factor, and where it binds to certain promoter regions and can be involved in gene transcription. Now, this other pathway here is where it would bind to this trimeric protein, where it can undergo several pathways that will activate expression of genes. And it's involved with a MAP kinase pathway. And it's important to note that this receptor is located on the endoplasmic reticulum, as compared to the straight nuclear pathway. And what we're really concerned with is whether activation through GPR1 is going to activate or turn on expression of any of these membrane progesterone receptors. And if it does, which one? But that's remained to be seen. The questions that we have mostly are even if it affects these levels, and if it does, we really would like to know what pathway and how the pathway functions as far as like the mechanism goes. This too is a mechanism of progesterone action in the brain, which is pretty similar to the previous slide. This P4 here is the type of progesterone that is most commonly found in the body. And this is similar to the classical nuclear pathway that we just talked about with estrogen, where P4 readily enters through the plasma membrane, binds with this progesterone receptor, and enters into the nucleus where it can bind to certain promoter regions and acts as a transcription factor. And the second pathway is where P4 binds to this membrane progesterone receptor, also known as MPR. And this is also a type of G protein coupled receptor, and they signal through a MAP kinase pathway, and it's more of an indirect pathway here, where it also ends up acting as a transcription factor in gene transcription. But there's more progesterone that can bind to the membrane progesterone receptors. And what we're interested in is how this is affecting the receptor levels and if there's an increase in progesterone by estrogen being present in the cell. This is also some work that was done by this group here in Mexico City. And to our knowledge, this is the only other group in the world that's working with signal transduction pathways initiated by the MPRs as compared to the classical nuclear pathway. And this is just a review of some of our methods and materials used. And this is our cell line here. Again, the CH157 human meningioma cells. So we culture them in this type of media here. And the cells were treated, once grown to the appropriate amount, they were treated with a certain agonist as well as an antagonist, as well as these types of estrogen receptors here, G15 and G36. They were also treated with a vehicle of DMSO, just used as a control. And the treatments were evaluated by toluene blue staining and with a proliferation and viability assay. This RNA was eventually isolated around 1 to 2 days after the treatment using this kit. And it was inspected, and today actually we are to this second step here where we're going to create some cDNA. And we have just gotten to this portion, and we're hoping to collect some more data. So hopefully you can join us again here in a week. And please ask some questions, and hopefully we'll have some more data for you to ask questions about. But please do, again, contact with us. And thank you for watching our video. And good luck on the rest of yours. Thank you.